we move on to this point that Dayspring makes over and over and over again in the series. He talks about how all cells make cholesterol. So this is important. So let's take a listen here.
Everybody talks of lipoproteins as if they're delivering cholesterol all over the place. That's what we have lipoproteins for. That's probably the last reason we have lipoproteins, because every cell in your body makes every cholesterol molecule it needs to do what it has to do, with a few rare exceptions. So if I'm a nose cell, I don't need some lipoprotein to come and deliver cholesterol to my cell, because I need cholesterol in my nasal cell membranes. That cell will make cholesterol. Every cell has the genetic power and the protein, the enzymology to make cholesterol to its heart's content.
And then later he says, I think I have it here.
Kidney or any place else, because those cells need cholesterol. Those cells will make it, or they'll get it by some free diffusion if they really need it, or absolutely virtually any cell of it absolutely needed cholesterol, because for some reason the synthesis was broken. Any cell could ultimately upregulate an LDL receptor, but most of them don't. So, because they have no need for the cholesterol that's in an LDL. Yeah.
All right. So as you can tell, he's quite passionate about this. And he's saying that every cell in your body makes every cholesterol molecule it needs to do what it has to do, with a few rare exceptions. And, if you need cholesterol, every cell has the genetic power, the proteins, and the enzymology to make cholesterol to its heart's content. And I took this from the transcript, so I know there are mistakes there. And then also, if it absolutely needed cholesterol because for some reason the synthesis is broken, any cell could ultimately upregulate an LDL receptor, but most of them don't. I spent a lot of time thinking about this, just because it tells me where I think the fuzzy thinking happened. So again, going back here, he's talking about the genetic power, the proteins, and the enzymology needed to make cholesterol. But if the synthesis part is broken, then they could then upregulate, these cells could then upregulate an LDL receptor, but most of them don't. So here you're seeing that he's separating synthesis from upregulation of LDL receptor. This is a paper from Arteriosclerosis, Thrombosis, and Vascular Biology, 2004. It looks at a transcription factor called SREBP2, or sterol regulatory element binding protein 2, mouthful, all right? And basically, what you need to know is this is a transcription factor that is involved in turning on cholesterol synthesis. So what it does is this transcription factor lives in the endoplasmic reticulum. When cholesterol levels drop in the cell, it becomes activated. It then travels to the nucleus and turns on genes, right, to upregulate cholesterol. We want more cholesterol, right? So now when it detects that there's low levels of cholesterol in the cell, it moves to the nucleus and turns on genes that will help it accumulate more cholesterol. And it does this by initiating the synthesis of cholesterol, very important, but it also is involved in expression of the LDL receptor. It says here, expression of the LDL receptor, a key component of the cellular pathway for cholesterol uptake, is modulated as a function of cellular cholesterol content through SREBP-mediated transcriptional regulation. So main points here, again, actually covered this on episode 46 of this podcast, but SREBP2 is very important for homeostatic control of cholesterol levels in the cell. Once activated, it goes to the nucleus and turns on genes for de novo biosynthesis of cholesterol and upregulates the LDL receptor. It does both things. It turns on the genes, a set of genes that do both. Okay, it doesn't just say, eeny, meeny, miny, mo, we'll just synthesize today. We won't do the upregulation, all right, unless the synthesis part is broken. So here you understand that you can't separate those two processes. It's not like, oh, we have the synthesis part and then we have the upregulation part and they are separated. That's not how your cell thinks. And, obviously, cholesterol synthetic pathways are quite a lot to, work with. They're very complex. There's many, many names here that you have to remember. So in part two, around the 40-minute mark, they go into what is cholesterol. They discuss cholesterol chemistry. And here, I just want you to look at the comments if you have a chance that were made by a viewer called Sobcheck2, who identified as an organic chemist. So this person made some comments in the YouTube version of this about their cholesterol chemistry. So I don't want to take credit for that. So I thought, I'll put it up there and you can take a look. And I think it's important because they themselves recognize how important it is to be accurate with the chemistry.
Know some of this stuff. Well, and that's the thing, you have to be willing to learn some of this chemistry. I mean, you have to steep yourself in biochemistry and understand because the significance becomes enormous. One double bond in one of these things completely changes its properties.
Absolutely. So I think because of the importance of this, that's probably why Sobchak 2 made those clarifications on YouTube. Right. So as I said, they talked a lot about how we make cholesterol synthesis, and they talk about the different pathways, the block pathway and the Kandach-Russell pathway. And it turns out that de novo synthesis of cholesterol is indeed highly, highly complex. There are many, many, many steps involved, at least 20 or so, regulated by numerous enzymes. And at one point, Peter Attia said, I remember learning this in med school, and it kind of brought me back to my own med school years. And I confess, I hated memorizing stuff for exams. So as they went on about the different enzymes and different points of the pathway, I was getting a little uncomfortable because I'm going to tell you now, I didn't do that. I was kind of lazy, and I just always tried to escape memorizing things. Actually have a very good memory, but I just hated memorizing stuff. Actually, you know what? I think Richard Feynman also hated rote memory, so maybe that's another thing we share. But I would just do whatever I could to not like memorize because I was that student. I was that kid that was always like, really? We have to memorize this? What's the point? Can't we just look it up, So it's a little bit whiny, I have to admit. I was lazy and I just didn't want to do the work. And then I would look at the pathway. I'd look at something like this, and my eyes would just glaze over, and I go, wow, that's a lot of work. That's a lot of work to memorize this. And then I think, wow, if it's so much work just to memorize it, I wonder how much work it would be for a cell to make this stuff. That looks like a lot of work. And then I would like go down rabbit holes, try to find this out and read and look things up because I just was procrastinating and just didn't want to study, right? So kind of waste time. And instead of studying, I'd do something like that. But it turns out that, interestingly enough, it takes a lot of energy to make cholesterol. To make 1 mole of cholesterol, you'd need 21 moles of NADPH, 18 moles of acetyl CoA, 36 moles of ATP. And if you remember that each, NADPH, is equivalent to 3 ATPs, when you run it through the electron transport chain, right? Then it turns out that you need over 100 ATP equivalents and 11 oxygen molecules to make one molecule of cholesterol. I mean, I'm exhausted just thinking about that's a lot of energy. So, if you were a cell and you had to get your supply of cholesterol, well, you could make it. You certainly could do de novo biosynthesis, right? So, if I were the cell, it'd be like, okay, yeah, let's make some dum dee dum dee dum. But I I I don't like cooking. I'm actually a decent cook, but it just seems like so much energy. So maybe here's my laziness showing again. But it seems like that's a lot of work to make it. So I might be more inclined personally to, think about outsourcing this and getting some takeout, right? So if you were a cell and you were trying to make cholesterol, increase your supply of cholesterol, for example, you could make some if you had, 100 ATP equivalents and 11 molecules of oxygen, just kind of, as spare change for one molecule of cholesterol. Or you might just save yourself some of that energy and, bring some cholesterol in from the environment. So my clarifications here are cholesterol homeostasis is regulated by the transcription factor SREBP2, which, when activated, upregulates de novo cholesterol synthesis and LDL receptor to bring in more cholesterol via LDL. It doesn't just turn on cholesterol synthesis only, right? You can't separate the two because it activates the genes that do both, right? And this is because cholesterol biosynthesis costs a lot of energy. As I said, over 100 ATP equivalents and 11 oxygen molecules per molecule of cholesterol made. So if you're a cell and you need cholesterol, right, you're always having to think about how much you have to spend, and you will probably might have to make some, but you'd also want to bring some in because it makes energetic sense to import most of your cholesterol rather than make it. And even pathogens, I note here, want to import it, right? So it's not just the cells in your body, but microorganisms in your body that might be invading you, right? Would be very interested in cholesterol because they need the cholesterol to make cell membranes and so forth. And so they are always looking for cholesterol. And it's almost a hide and seek game, as you'll see later on. I'll talk about this a little bit. It's almost a hide and seek game here because your body's trying to deprive those organisms of cholesterol while those organisms are actively seeking out the cholesterol, right? So it's not just the cells in your body, but microorganisms, pathogens, and even cancer cells. Okay. So I think that's where it's important to think about this carefully and separate out the fuzzy thinking, understand that you don't separate synthesis of cholesterol from upregulation of LDL receptor. Whenever the cholesterol levels in your cells drop, then your cell's priority is, I just got to get more cholesterol. And it's going to do whatever it needs to get that cholesterol. It would synthesize some, but it would want to bring some in because that's going to cost a lot less energy.